this. So what are monoclonal antibodies again? Right? We looked at the structures of antibodies before. They're generally the same thing, right? So the monoclonal antibodies, right, are different antibodies derived from a single clone of B cells, right? So the B cells will produce these antibodies, right? So they are specific to a single epitope within an antigen. We described all of those already. Unlike polyclonal antibodies, they are produced by different B cell linkages, right? And recognize multiple epitopes on the same antigen. The monoclonal antibodies are uniform in structure and specificity, right? So uniform in structure and specificity. Okay, that's important there. It's the general structure of the antibody. It will be the same thing, right? So how do we use them for diagnosis? Now, when we use them for immunoassays, right, monoclonal antibodies are widely used for diagnostic tests such as ELISA, right, an enzyme-linked immunosorbent assay, right, or assay. Lateral flow assays, and what we call the fluorescence activated cell sorting, right? So they're using these different ways, right? So they use the monoclonal antibodies in ELISA, right, are immobilized on a solid surface and used to detect the presence of specific antigens in patients, right? Samples by binding them together, right? So it's generally the same thing you use in a pregnancy test. No, same thing with the pregnancy test, but they now use different substances from the body and test it on those strips, and we're looking at how the antibodies bind to these antigens and how they move and the colors in which they present by the mobilization of the dyes. All right. We have pathogen detection, right? Specifically, so the monoclonal antibodies are employed in the detection of infectious agents, right? So our pathogens. So the rapid diagnosis tests for infectious diseases like HIV, malaria, and COVID-19 rely on monoclonal antibodies, right, to detect any type of bacterial or viral components. All right. So we have a detection of them there. So that's another important one, monoclonal antibodies. We also have blood typing. They're used in blood typing tests. So monoclonal antibodies are specific to ABO and RH blood groups, right? Antigens, and are used in blood typing, right? So blood tests, right? To figure out the patient's blood group. So those are three different uses for them in diagnostic uses. For treatment, now we have cancer therapy. So monoclonal antibodies are used in targeted cancer therapy, right? So they bind to selected cancer cells, right? Deliver cytotoxic agents, right, or block signaling pathways. So, like for example, MabThera, which is outlined in the syllabus, is a type of monoclonal antibody that is used to treat non-Hodgkin lymphomas or chronic lymphocytic leukemia, right? So, it were used to treat those type of cancerous agents. It targets the CD20 antigens on the B cells, leading to their destruction by the immune system, right? So looking at that, so leading to the destruction of these different types of cells, right? These cancer cells, right, by the immune system. Okay. So we're looking at these different things. All right. So the lymphocytic leukemia would actually help to destroy the immune system, etc. That's why leukemia is that dangerous, right? And our MabThera is used to neutralize these issues, right? These components, right, for cancer therapy there. Leukemia is cancer of the blood for anybody who never caught that a while ago. Autoimmune diseases as well. Our monoclonal antibodies can be used to modulate these responses, right? So they target specific molecules involved in autoimmune diseases, right? So, infliximab rather targets the tumor necrosis factor in alpha TNF. Alpha, right? And it's used to treat autoimmune diseases like rheumatoid arthritis and Crohn's disease, right? So it's used for that. So looking at different medications and how they're used for these different things, right? So they're antibodies that are used to treat these different issues. Infectious diseases as well, right? So monoclonal antibodies are used in also passive immunotherapy, right, to fight against different types of pathogens and to provide some amount of immediate immunity against pathogens, right? So palivizumab, right, is a monoclonal antibody used to prevent respiratory syncytial virus, right, RSV infections in high risk infants, right? So these are used of different antibodies. So some of the medications used in the treatment and diagnosis of these diseases are just antibodies, right? That we've looked at within the immune response. So tailored towards these different issues. As for pregnancy tests, now, how are antibodies used in pregnancy tests? We use the monoclonal antibodies, right, to detect human chorionic gonadotrophin, right? So we use these monoclonal antibodies to really detect these things. And once we recognize human chorionic gonadotrophin, that is usually a signal, right, that the person may be pregnant, right? The woman is pregnant, right? So it's usually present within urine samples, right, leading to visible signals on pregnancy tests. So the general principle, right? It uses LISA, right? As I stated before, enzyme linked immunosorbent assays, right? So it's basically a type of chromatography, right? Because they use an immunosorbent, right? Unit 2 students of chemistry would have understood what a sorbent is and stuff like that, right? So it's generally the same thing, right? So we have three monoclonal antibodies, right, that are specific to HCG, right? And it's conjugated to an enzyme that carries a dye, right? And the second monoclonal antibodies are specific to ACG but are immobilized, right? They're immobilized to the dye's substrate, right? So looking at that. So when the ACG is present in urine, it will interact with both sets of monoclonal antibodies, right? When both sets of antibodies are bound to ACG, the enzyme is brought into physical proximity to the dye, changing its color, right? The third set of monoclonal antibodies will bind to any unattached enzyme-linked antibodies, functioning as a control. So what's happening here? So the woman's urine is placed here, right? Step one, it's placed here on the test specifically, and we'll go through, right? The sorbent specifically, just go through, right? Same concept of the chromatogram, right? The HCG will bind to place monoclonal antibodies, right? Anti-HCG antibodies with enzymes that trigger color change. So they will bind now, right? Enzymes that trigger color change. Okay, you see that there. Step three now, already when we look at already placed anti-HCG monoclonal antibodies, they're going to now be binded to dye, right? So what's gonna happen here now? When these different complexes, right? HCG monoclonal antibody complex now forms, right? They're going to actually change color, right? So the monoclonal antibodies that are compatible, right, with the anti-HCG antibodies now bind and trigger a set of color changes. And this, right, what we're going to look at here, that would generally be the control over here, right? So if there's a color change here, what does that mean? If there's a color change there, that means that it's a positive test, right? If there's a color change here, that means the control is working. So this part will change no matter what, right? So in step four, it's the control. So the monoclonal antibodies for ACG are there, are already present on the slide. Therefore, a color change will occur no matter what, right? But in step three, the monoclonal antibodies are not present. So if there's a color change, right, due to monoclonal antibodies being present, then that will show that the monoclonal antibodies came from the urine. So what will happen here is that a line will come to show that there is a positive test, right? A positive pregnancy test, right? And then there will be a line after that will show that the test is actually working. Now, what would happen or what would generally occur, right? If a positive line were to be produced, but no line at the control, what do you think should be done?
Another test.
Another test, right? So another test must be done, right? And if the control is working and there is a positive line, then there is a possibility, right, of pregnancy, right? Or if the control shows, right, and there's no line at the test region, then it is said that there's a possibility, or there's majority of the times, then there's no pregnancy there, because there are also there are all types of false positives and false negatives that can occur. So it's usually the best bet would be going into a hospital for them to test the hormone specifically from your body, or to do a general ultrasound to know exactly whether or not pregnancy or gestation is occurring, right? But you guys look at ultrasound and all those type of things and the different types of uteral electrodes and stuff like that in Unit 1. So we're not going in there. Right? So it's generally just the structure and the function. How does the pregnancy test work? So we use the ELISA process, right? Enzyme linked immunosorbent assay, right? And it's basically chromatography. It's basically that. It's not really anything else, right? So this mobile phase will move up the stationary phase, and on the stationary phase we have two areas: a test area and a control area. The test area has receptors that bind to anti-HCG monoclonal antibodies. So if they're present, then it will show positive for the test area, right? And then on the control area, there are antibodies regardless, so it will show, right? So we have that there. If it's working, it will show. So that's really it for that part.